Hi, I'm Caroline Ganglo, a PhD student of System Link at the University of London, Koblenz. Welcome to this presentation on the effect of Bacillus thuringiensis israelensis on methane dynamics in ponds. There are several reasons to be interested in methane. First, it is the second most important anthropogenic greenhouse gas in terms of climate forcing after carbon dioxide. Atmospheric methane is a stronger absorber of the Earth's emitted thermal radiation, which is 28 times greater than that of the carbon dioxide. However, the surface dry air mode of atmospheric methane reached about 1,857 ppb in 2018, which, as shown in this graph, is almost 2 times greater than its estimated pre-industrial equilibrium value before the 1980s. This increase is attributable in large part to increased anthropogenic emissions of methane. Fortunately, methane has a shorter lifetime in the atmosphere. Therefore, stabilization or reduction of its concentration leads rapidly in a few decades to stabilize or reduce its atmospheric concentration and thereby reducing the global warming. Ponds are natural sources of methane emissions to the atmosphere. They are characterized by lentic waters, which means that the water bodies of these systems are not flowing. Globally, there are about 304 million of small ponds, less than 50 hectares, which cover about 4% of the land area. They comprise 98% of lentic waters by number and 33% of lentic waters by volume. Ponds are constructed because they provide essential ecosystem services such as water retention. They provide equipment with recreational activities. They are valuable habitat and resources for aquatic biodiversity, but they are also hotspot for the carbon biogeochemical cycle. Pond, for example, emits about 4 teragram of methane per year, a value which represents 41% of the global diffusive methane of lentic freshwaters. It is then crucial to understand the biogeochemical processes responsible for methane emissions in these shallow systems. As designated here in shallow system, organic matter settle to the sediment where oxygen is often absent. The settled organic matter are degraded by methanogenic archaea, which reduce acetate or carbon dioxide into methane. And then methane, once methane is produced, it is oxidized in the water column by methanotrophic bacteria, or part of the methane escape oxidation and are diffused to the atmosphere. When the production rate of methane in sediment exceeds its oxidation rate or transport diffusion rate, bubbles start to accumulate in sediment, and these bubbles are also transported directly from the sediment to the atmosphere at a high rate. Plants can also uptake methane from the sediment and then emit it to the atmosphere. And then the atmospheric carbon dioxide is needed by plants, which use it during photosynthesis to provide organic matter for the microbial decomposition, and the cycle of methane continues. The anoxic condition in sediment is often regulated by aquatic macroinvertebrates, namely the chironomid larvae. The chironomid are these ubiquitous species which represent 90% of aquatic macroinvertebrates. They basically, for feeding and respiration purposes, dig in sediment and thereby transport overlying oxygenated water through the burrow. This activity, known as bioturbation, subsequently changed the redox condition in sediment and increased sediment respiration, when it also enhances the exchange of solutes between the sediment pore water and the overlying water. Given this condition, what is the fate of methane fluxes in this system? On the other hand, BTI is the most environmental safe pesticide used as mosquito control agent during flooding, but it has some detrimental effect on the chironomid meats. For instance, mesocosm study proved that BTI reduces the chironomid abundance by 88% and then emergence by 50%. But we still don't know the effect of BTI on methane emissions in ponds. So we wanted to answer 2 questions. Does BTI affect net methane emissions in ponds? We expect BTI to increase methane emissions in ponds by reducing methane oxidative agents, such as the chironomid larvae. In this condition, we also expect methane oxidative rates to reduce in ponds. So to achieve our assumption, we sampled methane in these 12 ponds, which are at Enviromental in Germany. The figure A is the picture of the 12 pond. The numbered pond in yellow were the treated pond with BTI, whereas the intermediated pond without numbers here are the control ponds. In each pond, we identify two zones: an aquatic zone represented by submerged macrophyte, and aquaterrestrial zone represented by emergent plant. The pond surface area is about 148 meters square with a maximum depth of 0.5 meters. To cycle methane, we measure diffusion fluxes of methane in the aquatic zone of the pond using transparent chamber as shown in figure A. We subtracted evolution fluxes from the total fluxes, and we also measured plant-mediated transport of methane in the aquaterrestrial zone of the pond as shown in figure B. We calculate the headspace method was used to measure methane concentration in the surface water of the pond. We calculated methane oxidation by measuring the fractionation ratio of carbon 13 over carbon 12 in sediment bubbles and surface water of the pond using the stable isotope analysis. Then we continuously measure the water parameters and use linear mixed effect models as statistical tools. This diagram shows the experimental design of flooding and BTI application. So on the y-axis, we have the change in depth of the 12 pond over time. As indicated, before flooding the 12 pond, we measured methane concentration and fluxes, which were also taken after each flooding and BTI event. And also at each flooding and BTI event, we measure the same parameters. So there were 3 flooding and BTI application events, which happened between April and June. And then we continuously monitor the water. We continuously monitor the same parameters in the pond over the year to assess the BTI effect on methane dynamics, BTI post effect on methane dynamics in ponds. Over the year, the 12 ponds were methane saturated with average concentration of 2.8 micromole per liter in both aquatic and aquaterrestrial zones of the pond. But the highest concentration was observed in January of both zones, which reached about 1.5 micromole per liter due to the ice formation of the surface water of the pond that inhibited methane oxidation. The concentration range did not vary between BTI and control pond as indicated by this value. So BTI did not increase the response of methane concentration in both zones of the pond. But methane increased by 70% due to flooding for BTI application in spring, and also by 25% all over the year due to temperature, as shown here. The ponds were a net source of methane all the year, with an average diffusive methane emission of 1.8 millimole per square meter per day in the aquatic zone and 2.9 millimole per square meter per day in the aquaterrestrial zones. There was no significant differences between BTI and control pond during the sampling time in the aquatic zone, except in the aquaterrestrial zone in September, where BTI increased methane diffusion fluxes. Methane diffusion did not vary much in the aquatic zones of the pond, except from January, where we observed the high diffusion rate. But it does vary over time, as we will see in the aquaterrestrial zone. And this variation is 15% explained by temperature. So over the year, 67% of methane is oxidized in the water column of BTI pond, and 65% of the fraction of methane was oxidized in the control pond. Ebullition is the dominant pathway of methane emissions in the pond, which was followed by the diffusion fluxes. BTI affected methane emissions at one sampling period, which implies that BTI may have a long-term effect on methane emissions. However, this argument was not supported by our second hypothesis so BTI did not affect methane oxidation rate. This may be due to the large variability of processes controlling methane emissions, which may hide BTI's effect on methane emissions in open systems such as pond. This is why we need to repeat this experiment at a laboratory scale where environmental stresses will be reduced. We hope that controlling environmental stresses will increase our understanding of BTI effect on methane dynamics. Thank you very much for watching this presentation. I'm looking forward to discuss most of the point of this presentation with you. Thanks.